We're going to start our second session now. The first speaker is Cynthia Morton, who is a professor at Harvard Medical School. She's the president of the American Society for Human Genetics. She was the former editor, slight correction to what's in the bio, of the American Journal of Human Genetics. She's also the director of CYTOGENETICS at the Brigham and Women's Hospital. And Cynthia is going to talk about lessons about our genome from balanced chromosomal rearrangements.
Thank you. It's a truly special pleasure to be here for this grand opening symposium. I'm sure this will be the first of many symposiums, conferences, retreats, etc., that will take place here and that will propel human genetics and continue the many contributions of the Jackson Laboratory of Genetics in understanding health and disease. As I was sitting here, I was thinking that this is sort of a circle for me in that, which began in mapping genes from a conversation at the Jackson Laboratory short course in Bar Harbor in 1981, which led within that year to the assignment of the immunoglobulin heavy chain gene to 14q32 and the MYC gene to 8q24. And it did not escape our attention that the 814 translocation in Burkitt lymphoma was likely due to the liaison of these two genes. And today, what I'll tell you about has been sort of a journey of trying to understand chromosomal rearrangements and use them to annotate the human genome. And certainly before the Human Genome Project, each translocation or rearrangement that was deciphered was the subject of an entire R01, and of course all of that changed and has changed dramatically just in the past few years with the implementation of next generation sequencing. So the project I'm going to tell you about we call DGAP. It's for the Developmental Genome Anatomy Project, and the hypothesis of the project is one that's a paradigm in human genetics, that chromosomal rearrangements in individuals with congenital anomalies can be etiologic in the abnormal phenotype due to disruption or dysregulation of genes that are critical in human development. Now, we all know now that we don't have to look under the lamppost to find the keys or the genes, but I can assure you that in this project, all the keys are right there under the lampposts. And this map, which we like to refer to as the DGAP gene map, shows the locations of genes that, through studying chromosomal rearrangements in individuals with clinical phenotypes, has resulted in their disruption or dysregulation. And the ones that are indicated in blue type were in the first part of this project, and then you can see the amazing change in the number of assignments that occurred through the use of next generation sequencing to directly sequence the rearrangements, which before had been done by successive fish mapping, southern blotting, cloning the rearrangements, and Sanger sequencing. So it's really been now possible to do these in a very short time frame. And I'm going to talk to you about 4 sort of little stories that relate to this project, and one really makes the time frame issue important, and that's number 3, prenatal diagnosis. So using this technology to make diagnoses in times that will allow some action to be taken. So our method is to use jumping libraries. So we're using next generation sequencing, but libraries that have a bit larger inserts, 2 to 3 kilobases, and looking for ones that span a chimeric breakpoint. So if you think of the green signal as being on one chromosome and the red signal on another, these are sequences that shouldn't be together in the genome. And since we know the rearrangement that's present in the individual and for whom that library has been made, it's possible for us to get clusters of these fragments and then sequence them and confirm them then to nucleotide level resolution. So I'm going to tell you one of my favorite stories, but there are lots of stories and lots of lessons from the cases. And the first story that I'm going to tell you in gene discovery and genome annotation actually relates to my interest in the genetics of hearing and deafness. And among the PIs in the project, we all have our special interests, and that is one of mine, and I tend to pick off those cases to really study. So this was a young man who had profound sensorineural hearing deafness from a child, and had some mild craniofacial anomalies that you might appreciate from his picture. Interestingly, while he was a participant in our study, and we were really focused on trying to understand how his chromosome rearrangement that disrupted or that involved chromosome 2 and 13, how that might account for his hearing loss, he sent me an email to say that he had been diagnosed with prostate cancer at age 38, and he wondered whether his chromosome rearrangement had anything to do with that. I wondered too. I was a little, frightened to say yes or no, because basically I didn't know. So I said, I have no idea. So what we did was, and this case was started before we actually had next generation sequencing. Turns out that his translocation disrupted an unannotated sequence known as C2ORF43. And this was located on chromosome 2. It broke this gene between exons 5 and 6. It resulted in 2 abnormal transcripts from abnormal splicing products and haploinsufficiency, about a 50% decrease in the normal transcript. And you can see here that the rearrangement, when analyzed at nucleotide level, resulted in an insert of 68 base pairs of untemplated sequence, so sort of a little bit of noise at that breakpoint. And then given below is what the way we are now describing these chromosomal rearrangements. It's a full employment act for cytogeneticists. But I will show you a little bit at the end how this whole project in studying these individuals has led us to develop a new nomenclature that will do what cytogenetic nomenclature has done for many, many years now, which is to provide an interoperable nomenclature that we can describe rearrangements and understand exactly what they mean, no matter what area of the globe one might reside. So the hearing loss, again, was my interest, and what you can see is that when we analyzed the hearing in about 12-month-old mice, and the boys are in blue and the girls are in pink and red, we found that there was a significant threshold increase in the male mice, and interestingly, not a significant increased threshold in the female mice. And we know that men actually have a greater frequency of hearing loss than women, and about 1 in 3 individuals at age 65 has age-related hearing loss, and it's more common in men than women. But we also know that over the time that people have studied age-related hearing loss, certainly men have been involved in occupations that were more likely to cause noise trauma. But interestingly, that in fact, we found this by stratifying the mice by sex. And while we were continuing to try to understand this, a paper appeared in Nature Genetics of a GWAS for susceptibility for prostate cancer in Japanese population. And the top, the gene listed in the red box is C2ORF43. So obviously this was of intense interest to us, and we began to try to understand a bit more about how this unannotated gene might play a role in prostate cancer. And one thing we did, and this shows sort of the ability to use all kinds of convergent genomic evidence these days, It turns out that this gene's expression in primary and metastatic prostate tumors is very significantly downregulated as compared to a list of genes, cosmic genes that are recognized for prostate cancer. So you can see the C2ORF downregulation in the primary and in metastatic tumors. Then what happened with the mice? So it turns out that we have an increased frequency of abnormal prostates in our heterozygotes and knockout mice, and including some mice that appear to have microinvasive and invasive prostate tumors. So it appears that this gene has a role that we will be able to, as time goes on, perhaps even implicate more in prostate tumorigenesis, and we'll be able to use this N of 1 experiment to help us understand hearing loss and prostate tumors. But that's not all. We happen to be told that we had the fattest mice in the animal room. And it turns out, I think you can see the difference in the size between these two litter mates, the wild type litter mate and the knockout female. So the fat mice are the female mice, and you can see that by about 8 months, they begin to really differentiate from their male sibs. So this gene appears to have a role that is also related to the sex of the mouse. So what do we know about this gene? Well, it turns out not too long ago, it's been determined to be a cholesterol esterase, and it cleaves fatty acids from cholesterol at the surface of lipid droplets. So there's quite a few different hypotheses that you can make, and certainly in terms of its role in hearing, there's been an interest in cholesterol and lipid dysregulation associated with hearing loss. So this has really opened up sort of an oncometabolic pathway for us that we never knew that we were going to be in, but that's what these cases do. These are real human, this is real human biology. It's often more complex than you recognize, but it can give you great insight into different genes. So that's just a short, short gene discovery story. And now I'll just tell you a little bit about what we've learned about genomic structural rearrangements. It turns out that in sequencing about 100 of these, we found that not only did we disrupt genes, we also disrupted non-coding RNAs. And I think the total number out of our 131 cases with 176 disrupted genes, we have 19 non-coding RNAs disrupted. We thought by the karyotype analysis that about 3% of the rearrangements could be categorized as complex, meaning there were more than 2 breakpoints. But at base pair resolution, we find that about 20% are complex rearrangements. So greater than 3 junctions per rearrangement. And you can see here that in just 2 cases, 2 of the first cases that we really studied to try to put back together, we found that a mechanism that had been recently described in cancer of chromothripsis, where there's a massive shattering and reorganization, also took place in the germline. And so we have a number of these cases now. And one of them involved 14 junction fragments from 2 shattered regions of chromosome 5, and the other one on this slide shows 11 junction fragments from 3 shattered regions of chromosome 7. So what's the difference then between what we see in cancer and what we see in the germline? Well, I think the difference is that in the germline, we're really not very unbalanced. We've seen a few base pair losses, I mean, in the low KB range, but not the real massive aneuploidy that you see in cancer. And probably that reflects the fact that these are individuals who have survived and are on the delivery table, so probably could not accommodate really the massive aneuploidy that accompanies chromothripsis in neoplasm. So just to show you, this reminds me to tell you that almost every time when we sequence the rearrangements to nucleotide level, we end up revising the karyotype. Perhaps that's not a big surprise. I think that cytogeneticists have done a great job over decades of looking down from the 30,000 mile high range and trying to assign a breakpoint, but in fact it's just impossible. And because these rearrangements are often more complex than we thought, it also adds some difficulty in trying to call the breakpoint. In this particular case, there was about 6 kb bases deleted out of all the 11 rearrangements. And just to show you what we saw in terms of how the pieces were swapped and knitted back together, it's really a complex story. This is our dancing chromosome. And in another case, the other case that was on that slide, I won't take you through the dance of that one, but it involved a chromosome that we didn't even know was involved in the rearrangement. The rearrangements had been called as a 3-5 rearrangement, and it turned out there was not a single junction fragment that actually joined a piece of 3 to 5. There was also chromosome 7 involved in that rearrangement as well. So what we've also learned in studying and thinking about the structural rearrangements is that these rearrangements are really non-homologous in-joining rearrangements as compared to the types of rearrangements that we're familiar with from copy number variant rearrangements that are recurrent, that include microhomology. There's really not evidence of microhomology at these breakpoints. That doesn't mean there's not some higher order genomic structure of the chromosomes that are involved in these rearrangements that actually precipitates their translocations. So I'll move on to the little story of the precision prenatal diagnosis. We have now completed 6 pregnancies, 6 cases where we resolved the cases to nucleotide level resolution. All of these were referred for advanced maternal age or an abnormal screen. They all had balanced, apparently balanced de novo rearrangements. And we know from work of Dorothy Werber in the early 1990s that the risk of an untoward outcome from a de novo rearrangements picked up in prenatal analysis is stated to be 6.1% for a translocation and 9.4% for an inversion. And I've heard from the group in Denmark, from Niels Tommerup's group, that they believe these are actually a bit underestimates, and it's really around 20% when they follow the child out from the delivery. Interestingly, all of these cases, and we have 2 others in progress right now, had normal array CGH. So these translocations or inversions were not picked up by any array. And we've been able to then take them all to, except for the 2 that are currently underway, which I hope will be successful, to nucleotide level precision. So here's the first case. This is a 41-year-old. This was her 3rd pregnancy. She had 1 normal living child. It was 16 weeks. She had normal first trimester screening, and I was really blessed. This is a woman who is also a scientist and spends her life cloning mouse translocations. So she was right on the browser with us through this whole process, and it was really, really wonderful in that way. She was the 2nd case that we did, and it is a totally white knuckle experience. So she had an inversion, or her child had an inversion in chromosome 8. You can see the original breakpoints that our cytogenetics lab assigned, and then after sequencing a revision of those breakpoints, turns out that a gene known as KHDRBS3, which is an RNA binding protein, is disrupted at one breakpoint, and the other breakpoint is in a non-genic region. The green shading on the ideogram is the region that would have been designated by the cytogenetic resolution, and of course, once we go to nucleotide level, you see this straight red line through there exactly telling you where the breakpoint was. And we're really happy that based on all the convergent genomic evidence that we could look at that point, she and her husband decided to continue the pregnancy. And at 23 months, the child is doing well, is within normal limits. And at 18 months, she made this little clip for us this is a clip from a much bigger video, but every time I hear her say these words, I feel we live in a really special time now. So let's.
If we were not here, we were in some other place, most likely Julian would not be born.
So she says that most likely her son would not have been born if we hadn't been able to tell her, based on the evidence that we thought that there was no that it was a variant of unknown significance, but that it seemed that it could be consistent with normal development. So it's sort of amazing that somebody tells you this. So the second case I'll just tell you about, and I picked them for various reasons. This was a 27-year-old with an IVF pregnancy at 16.6 weeks. She had an abnormal maternal serum screen. I wasn't happy to find out there were twins, but anyways, the twins had a SNP array, and we were able to, because they were determined to be monozygotic by the SNP array, we were able to combine their DNA samples that were left over from the array CGH and get right to the sequencing. There was an inversion in chromosome 6, and again, a revision of the breakpoints, and I think the best diagnosis that you can get from us at this point is that both breakpoints were in non-genic regions. The twins were normal at birth. But what was also interesting, as you can see that little purple band at the top, we picked up an inversion that we didn't know about and was really cryptic to us from the cytogenetic analysis. So it gave us a belief that, in fact, we don't have to know where we are to see rearrangements and pick them up. So it turned out that that was a paternally inherited inversion. And of course, we didn't determine which chromosome it was on, whether it was on the chromosome that included the inversion, the large inversion, or not. But it was outside of the region of the chromosome inversion that we were sequencing. Then we had another case that was a 44-year-old at 17 weeks. This pregnancy ended in a termination, a natural termination at 23 weeks due to high drops. There had been an abnormal nuchal translucency, but normal 15 and 22-week fetal scans. This one involved chromosome 10 inversion. You can see again the revision of the karyotype. It interrupted a gene known as CPEB3, which is a cytoplasmic polyadenylation element binding protein. But in this case, we had another inversion at the rearrangement site, and it just reminds me to tell you that, in fact, we found in sequencing the cases over the life of this project that often it's not unusual to find that there's an inversion at one of the chromosome breakpoints. So probably also part of the mechanism for these rearrangements. So the last story is just to tell you a little bit about what we've done in trying to put together a next-gen cytogenetic nomenclature, which was really a journey with one of my postdocs. And when we finally submitted the paper for publication, it was version 78, and there was a 115-page supplement. So there is a committee called the International Standard in Cytogenetic Nomenclature, and this is the Bible for cytogenetics in terms of defining rearrangements. And I have to say we're very careful to title our paper for a suggested next generation cytogenetic nomenclature. The current rules give guidance on microarray nomenclature and region-specific assays, but now we will move to sequencing, and there will be a meeting the end of October of the committee to address various issues, including providing nomenclature for a nucleotide level resolution. So this is sort of what it looks like, two chromosomes on their side that are involved in a rearrangement, and really providing not just the band, but exactly the nucleotide. And we have a short system, and then we have provided sort of a longer term that's also a little bit shorter system to indicate also the fact that this is from a sequenced rearrangement and what builds of the genome are involved. Now, this is not a trivial thing to do, so we have worked on a program which we call Boston, and the reason we call it Boston, not only because the acronym fits really well for Blast Output Sequence Tool of Nomenclature, but there have been some really historic meetings of cytogeneticists starting in Denver and then London and then Chicago and Paris, so we decided let's try to do this as our Boston program. So you can put the sequence in across the breakpoint and it will spit out the nomenclature. So there are lots of people that I have to thank for this work, but especially the families. And I think here you can see what I sometimes call the faces of DGAP. These are all N of 1 cases where they don't have a support group because their rearrangement is not something that has been seen previously, and no one is able to give them very much news. So this sequencing project has really allowed us to provide information, and I can't tell you how heartwarming responses we've received from parents who it's totally true that a real diagnosis is therapeutic in itself, even if there's nothing that can be done. And then I have lots of some wonderful colleagues, counselors, graduate students, postdocs, and clinical geneticists all around the world who contributed these cases. We have over 300 cases now, and I really think we got to grab all of these cases because they annotate the genome in some ways in a very inexpensive way, and have given insight to some things that we would have never anticipated. So thank you very much, and again, congratulations to Jackson Auditory.